Photosynthesis. 2.1 Discovery of photosynthesis. The history of the studies done on photosynthesis dates back into the 17th century with Jan Baptist van Helmont. He rejected the ancient idea that plants take most of their biomass from the soil. For the proof, he performed willow tree experiment. He started with a willow tree of 2.27 kg. Over 5 years, it grew to 67.7 kg. However, the weight of the soil only decreased by 57 grams. Van Helmont came to the conclusion that plants must take most of their weight from water. He did not know about gases. Joseph Priestley ran a series of experiments in 1772, Fig. 2.1. He tested a mouse, a candle, and a sprig of mint under hermetically sealed, no air can go in or out, jar. He first observed that a mouse and a candle behave very similarly when covered in that they both spend the air. However, when a plant is placed with either the candle or mouse, the plant revives the air for both. Further ideas were brought about in the late 1700s. Jan Ingenhuys and Jean Seen-Beer found that the air is only reviving in the daytime and that CO2 is assembled by plants. Antoine Laurent Lavoisier found that revived air is a separate gas, oxygen. But what is the oxygen maker? There are many pigments in plants, and all accept and reflect some parts of rainbow. To identify the culprit, Thomas Engelmann ran an experiment, Fig. 2.2, using a crystal prism. He found that Spirogyra algae produce oxygen mostly in the blue and red parts of the spectrum. This was a huge find. It tells that the key photosynthetic pigment should accept blue and red rays, and thus reflect green rays. Blue-green chlorophyll best fits this description. Another important fact was discovered by Frederick Blackman in 1905. He found that if light intensity is low, the increase of temperature actually has very little effect on the rate of photosynthesis, Fig. 2.3. However, the reverse is not exactly true, and light is able to intensify photosynthesis even when it is cold. This could not happen if light and temperature are absolutely independent factors. If temperature and light are components of the chain, light was first, ignition, and temperature was second. This ultimately shows that photosynthesis has two stages. The first is a light stage. This stage relates to the intensity of the light. The second stage is the enzymatic, light-independent, stage which relates more with the temperature. Light reactions depend on the amount of light in water. They produce oxygen and energy in the form of ATP. Enzymatic reactions depend on carbon dioxide and water. They take energy from the light reactions and produce carbohydrates. Sometimes, enzymatic stage is called dark, but it is not correct because in darkness, plant will run out of light stage ATP almost immediately. Only some C4-related processes, see below, could run at night. Since water molecules are spent on light stage to make oxygen and at the same time are accumulating, see below, one of the best equations describing photosynthesis as a whole is CO2 plus H2O plus light right pointing arrow carbohydrates plus H2O plus O2. 29. Light from prism. Figure 2.2. Experiment of Th. Engelmann, 1881. 2.2 Light stage. The light stage participants include photosystems, chlorophyll, light, water, at pace, protons, and a hydrogen carrier, NADP+. The basic idea of light stage is that the cell needs ATP to assemble, later, carbon dioxide into sugar, Fig. 2.4. To make ATP, the cell needs electrical current, proton pump. To make this current, the cell needs the difference of electric charge, difference of potentials, between thylakoid, vesicle or membrane pocket, and matrix, stroma, compartments of the chloroplast, Fig. 2.5. To make this difference, the cell needs to segregate ions. Positively charged go from outside and stay inside, negatively charged go from inside to outside. To segregate, the cell needs the energy booster sun rays caught by the chlorophyll molecules embedded in the thylakoid membrane. The chlorophyll molecule is nonpolar, similarly to membrane lipids, and contains magnesium, Mg. It is easy to excite the chlorophyll molecule with light. Excited chlorophyll may release the electron if the energy of light is high enough. To make carbohydrates from carbon dioxide, CO2 apparently has no hydrogen. The cell needs hydrogen atoms, H, from hydrogen carrier, NADP+, which at the end of light stage becomes NADPH. The main event of the light stage is that chlorophyll reacts with light, yielding electron, E, and becoming oxygenated, positively charged molecule. Then electron, Pro. 30. Photosynthesis. 30 degrees Celsius. B. 20 degrees Celsius. Light intensity. Figure 2.3. Explanation of experiment of F. Blackman, 1905. A low light, little temperature effect. B. High light, significant temperature effect. TUN and NADP plus react to yield NADPH, which will participate in enzymatic reactions later on. The positively charged chlorophyll is extremely active chemically, therefore it splits water molecules, photolysis of water, into protons, which accumulate inside thylakoid, oxygen, O2, and electron. The electron returns to chlorophyll. When increasing gradient reaches the threshold, the proton pump starts to work as protons, H, pass along the gradient. The energy of passing protons allows for the ATP synthesis from ADP and P, inorganic phosphate. On the other side of membrane, these protons make water with hydroxide ions. In the previous paragraph, chlorophyll is actually two photosystems, photosystem II, P680, and photosystem I, P700. Photosystem II contains chlorophyll and carotenes, is more important. It splits water, makes proton the gradient and then ATP, and forwards electrons to photosystem I. Photosystem I contains only chlorophylls and makes NADPH. Ultimately, the light stage starts from light, water, NADP plus, ADP, and results in an accumulation of energy, ATP, and hydrogen, NADPH, with a release of oxygen, which is a kind of exhaust. Photosystems. Segregation of ions. Difference of potentials. Proton pump. ATP. CO2 assimilation. Figure 2.4, the logical chain of light stage reactions, hydrogen carrier not shown. 2.3 Enzymatic stage. The enzymatic stage has many participants. These include carbon dioxide, hydrogen carrier with hydrogen, NADPH, ATP, ribulose biphosphate, RUBP, or C5, and rubisco, along with some other enzymes. Everything occurs in the matrix, stroma, of the chloroplast. The main event of the enzymatic stage is CO2 assimilation with C5 into short-living C6 molecules. Assimilation requires rubisco as an enzyme. Next, this temporary C breaks into two C3 molecules, PGA. Then, PGA will participate in the complex set of reactions which spend NADPH and ATP as sources of hydrogen and energy, respectively, and yields through the intermediate stage of PGAL, 1 molecule of glucose, C6H12O6, for every 6 assimilated molecules of CO2. NADP plus, ADP and P, will go back to the light stage. This set of chemical reactions returns RUBP, which will start the new cycle of assimilation. Consequently, all reactions described in this paragraph are part of the cycle, which has the name Calvin cycle, or C3 cycle, because the C3 PGA molecules here are most important. In all, enzymatic stage starts with CO2, NADPH, ATP, and C5, RUBP. It ends with glucose, C6H12O6, NADP plus, ADP, P, and the same C5. With an addition of nitrogen and phosphorus, glucose will give all other organic molecules, Fig. DNA. Matrix, stroma. Figure 2.5, chloroplast. To summarize, the logic of photosynthesis, Fig. 2.8, is based on a simple idea: make sugar from carbon dioxide. Imagine if we have letters S, G, U, and need to build the word sugar. Obviously, we will need two things, the letter R and the energy to put these letters in the right order. The same story occurs in photosynthesis. It will need hydrogen, H, which is the absent letter, from CO2 because sugars must contain H, O and C. NADP plus NADPH is used as hydrogen supplier, and energy is ATP, which is created via proton pump, and the proton pump starts because light helps to concentrate protons in the reservoir. Rubisco is the enzyme of extreme importance since it starts the assimilation of carbon dioxide. Unfortunately, rubisco is two-faced, since it also catalyzes photorespiration, Fig. 2.9. Photorespiration means that plants take oxygen instead of carbon dioxide. Rubisco catalyzes photorespiration if there is a high concentration of oxygen, which usually is a result of intense light stage. Rubisco oxygenates C5, RUBP, which turns into PGA and PGAL, becoming glycolate. This glycolate is returned to the Calvin cycle when the cell uses peroxisomes in mitochondria and spends ATP. The process of photorespiration wastes C5 and ATP, which could be more useful to the plant in other ways. ATP. RUBP. Carbon dioxide. RUBP. ATP NADPHCO. Normal C, cycle. Photorespiration. Rubisco. PGA. Sugars. PGA. PGAL-PGA. Carbon dioxide. ATP NADPH. ATP NADPH. Figure 2.9, rubisco is two-faced enzyme. If concentration of CO2 is high enough, assimilation will overcome photorespiration. Consequently, to minimize the amount of photorespiration and save their C5 and ATP, plants employ Le Chatelier principle, equilibrium law, and increase concentration of carbon dioxide. They do this by temporarily bonding carbon dioxide with PEP, C3, using carboxylase enzyme. This results in C4 molecules, different organic acids, like malate, malic acid, with 4 carbons in the skeleton. When plant needs it, that C4 splits into pyruvate, C3, plus carbon dioxide, and the release of that carbon dioxide will increase its concentration. On the final step, pyruvate plus ATP react to restore PEP. Recovery of PEP does cost ATP. This entire process is called the C4 pathway, Fig. 2.10. Plants that use the C4 pathway waste ATP in their effort to recover PEP, but they still outperform photorespiring C3 plants when there is an intensive light and/or high blue temperature and consequently, high concentration of oxygen. This is why in the tropical climate, C4 crops are preferable. 2 groups of plants use the C4 pathway. Many desert or dryland plants are CAM plants which drive the C4 pathway at night. They make a temporal separation between the accumulation of carbon dioxide and photosynthesis. CAM plants make up 7% of plant diversity, and have 17,000 different species, for example, pineapple, ananas, cacti, cactaceae, jade plant, crassula and their relatives. Classic C4 plants drive C4 pathway in leaf mesophyll cells whereas their C3 is located in so-called bundle sheath cells. This is a spatial, rather than temporal separation. These C4 plants make up 3% of plant biodiversity and have more than 7,000 different species, for example, corn, sorghum and their relatives. In all, both variants of C4 pathway relate with concentration of carbon dioxide, spatial or temporal, Fig. 2.11. Both are called carbon-concentrated mechanisms or CCM. There are plants which able to drive both C3 and C4 pathways, like autograph tree, clusia, and plants having both classic C4 and CAM variants, like portulacaria. CO2 plus H2O plus energy carbohydrates plus O2. However, as most eukaryotes, plants have mitochondria in cells and use aerobic, oxygen-related, respiration to obtain energy. Carbohydrates plus O2 CO2 plus H2O plus energy. Typically, plants spend much less oxygen in respiration than they make in photosynthesis. However, at nights plants do exactly the same as animals and make only carbon dioxide.